Dear students, welcome to this lecture. Today, my topic is soil nitrogen, forms and factors affecting availability. Nitrogen is biologically the most significant element. It's a vital plant nutrient which is the constituent of two most preeminent biological compounds, that is nucleic acids and proteins. Of the three primary nutrients, which include nitrogen, potassium and phosphorus, nitrogen is required in the largest quantities. Nitrogen promotes rapid growth, increases leaf size, hastens crop maturity, and promotes fruit size and seed development. Nitrogen plays a role in almost all the metabolic processes in plants. Being an important constituent of amino acids, it's present in enzymes which regulate metabolism. Nitrogen is an integral part of chlorophyll molecules and is present in its spiral rings. Nitrogen constitutes about 78.09% of atmosphere by volume, yet plant growth in most of the terrestrial ecosystems is limited by non-availability of this nutrient. The problem is not the deficiency of this nutrient per se, but its non-availability in the forms in which it's taken up by plants. Nitrogen cycle involves complex relations between different forms of nitrogen in different oxidation states, from +5 in nitrate to -3 in ammonia. Living organisms, especially bacteria, have a far greater involvement in nitrogen cycle compared to the cycling of any other nutrient. Nitrogen fixation forms the main gateway through which inorganic nitrogen enters the biological systems. Other important processes which influence the labile soil nitrogen pool include ammonification, nitrification and denitrification. Now we'll discuss these processes one by one. So first is the nitrogen fixation. The process of combining atmospheric dinitrogen with hydrogen to form ammonia is known as nitrogen fixation. Nitrogen fixation is chiefly carried out by prokaryotic species. Such organisms which are capable of utilizing atmospheric dinitrogen as their nitrogen source are known as diazotrophs, and this kind of nitrogen nutrition is known as diazotrophy. Nitrogen is fixed by physicochemical, electrochemical and biological methods. Biological nitrogen fixation by far is the most significant method of nitrogen fixation, contributing 54 × 10⁻⁶ metric tons of fixed nitrogen per year to soil nitrogen pool. The other two methods jointly contribute only 7.6 × 10⁻⁶ metric tons of bound nitrogen per year to soil nitrogen pool. Haber-Bosch process, which is used in manufacture of synthetic nitrogen fertilizers, contributes 30 × 10⁻⁶ metric tons of nitrogen to fixed soil nitrogen pool. Now we'll talk about biological nitrogen fixation. Biological nitrogen fixation is divided into two categories. First is the symbiotic nitrogen fixation and second is non-symbiotic or asymbiotic nitrogen fixation. Symbiotic nitrogen fixation mostly occurs in the terrestrial systems and involves mostly bacteria, but also includes fungi and algae. Symbiotic association between members of Leguminosae and Rhizobium species is the most well-known nitrogen fixing association. However, symbiotic nitrogen fixation is not restricted to Leguminosae only. Many non-leguminous plants have been reported to harbor prokaryotic organisms capable of fixing atmospheric nitrogen. Examples include Myrica and Alnus species, which harbor nitrogen-fixing actinomycete, Frankia, in their roots. The plants entering into symbiotic association with actinomycetes are known as actinorhizal plants. Cyanobacteria form a symbiotic nitrogen-fixing association with fungi in lichens, while as Paspalum notatum from Poaceae encourages associative nitrogen fixation. The establishment of legume rhizobium symbiosis is a complex process involving physiological and biochemical properties of both the bacterium and the host plant. The development of symbiosis requires a coordinated expression of a number of plant and bacterial genes. Formation of visible nodules on root hairs is preceded by mutual recognition of a plant host and rhizobium species. Development of symbiosis involves intricate signaling network in which quorum sensing has been implicated as a key player. Bacteria are concentrated in and around the plant's roots, and formation of root nodules takes place as a result of proliferation of cells of root cortex triggered by increased supply of hormones from the bacterial symbiont. Mere presence of nodules does not indicate active symbiosis. Now, the non-symbiotic nitrogen fixation. It occurs both in terrestrial and aquatic systems and involves bacteria as well as algae. Among symbiotic nitrogen fixing organisms, Clostridium is the best characterized one. It fixes a nitrogen without entering into symbiotic association with plants. Now, the process of nitrogen fixation. Atmospheric dinitrogen being held by a triple bond between its two nitrogen atoms is chemically a stable molecule. The breaking of this triple bond consumes 160 kilocalories per mole. The union of hydrogen with nitrogen to form ammonia releases 13 kilocalories per mole, bringing down the energy input for nitrogen fixation to 147 kilocalories per mole. Nitrogenase complex, the enzyme system involved in nitrogen fixation, solves the energy requirement problem for nitrogen fixation. Nitrogenase complex is strictly a prokaryotic enzyme complex and has not been reported from any eukaryote till date. It's highly oxygen sensitive and requires to be shielded from oxygen in aerobic systems. Therefore, most of the algae carry out nitrogen fixation in special cells known as heterocysts, and legumes synthesize leg hemoglobin to ensure safety of nitrogenase complex. Nitrogenase complex consists of two enzymes, nitrogenase, which is also known as a component first, and nitrogenase reductase, also known as a component second. The former is a tetramer and contains molybdenum, while as the later is a dimer. Molybdenum of nitrogenase under the conditions of molybdenum deficiency in some species can be replaced by vanadium. Nitrogen fixation is a reductive process in which nitrogen passes from oxidation state of 0 to -3. 8 electrons are required to reduce one molecule of nitrogen to a molecule of ammonia. Of these, 6 are used for reducing nitrogen to NH3, while 2 others are evolved as hydrogen gas. For each electron transferred, 2 ATP molecules are consumed. Thus, 16 ATP molecules are consumed per molecule of nitrogen fixed. Some organisms produce an enzyme that is known as uptake hydrogenase to partially salvage energy wasted in production of H2. Nitrogen fixation is optimum when a nitrogen level in soil is low. This is true for both free-living as well as symbiotic nitrogen fixing systems. Nitrogen, when applied as fertilizer, tends to reduce the efficacy of symbiotic association. Thus, soils with relatively higher nitrogen levels may not support nitrogen fixation strongly. Now, the next process is ammonification. Ammonification is also known as mineralization. It refers to the decomposition of organic nitrogen-containing substrates like human and animal excreta, remains of dead plants and animals, and bacterial cells to release NH4 from them. Ammonification is an energy-releasing process. The energy released during ammonification is used by ammonifiers in maintaining their life processes. Ammonium is the preferred nitrogen source for many organisms, though some bacteria and fungi assimilate nitrate preferentially. Ammonium binds to cation exchange sites in soil and remains readily available for plant uptake. However, it may be immobilized in the interlayers of soil clay particles such as vermiculite. Many heterotrophic bacteria, actinomycetes, and fungi in soil and water use the organic nitrogen-rich substrate and return the inorganic nitrogen to the soil. Approximately 10 raised to the power 5 to 10 raised to the power 7 bacteria in 1 gram of soil are active nitrogen mineralizers. While some of these mineralizers are able to use a wide variety of organic compounds as their substrates, others are more substrate specific and act only on a particular substrate. During mineralization, complex polymers are first broken down into their simpler constituents, which are then acted upon by different enzymes to effect mineralization. For example, chitin is broken down into its constituent N-acetyl glucosamine units, which are further decomposed. Similarly, proteins are broken down into peptide chains, which are decomposed into individual amino acid units. These subunits may be used as building blocks for synthesizing bacterial proteins or may be mineralized to release ammonia. Carbon skeletons are usually kept intact and used in building biomass. Nucleic acids in the soil are acted upon by nucleases, which may either attack the nucleic acid chain at its ends or in the middle. Nucleotides released from nucleic acids produce urea upon their degradation, which in turn produces ammonic nitrogen. Now, the next process is nitrification. The process of microbial oxidation of NH4 and organic nitrogen into nitrate via nitrite is known as nitrification. Nitrification proceeds rapidly in warm, moist and well-aerated soils. Nitrification adds nitrate to soil, which is the chief source of plant nitrogen nutrition. Nitrate is one of the most common forms of nitrogen in the environment. Theoretically, the oxidation of atmospheric nitrogen to nitrate is favorable. However, due to the occurrence of triple bond between two nitrogen atoms in a nitrogen molecule, this conversion does not occur in nature very readily. Lightning provides enough energy to favor this transformation and contributes anything between 0.5 to 5.0 kilograms of nitrate nitrogen per hectare per year to soil. Apart from nitrification, there are many other sources of nitrate in the soil, which include atmospheric deposition and addition of nitrate and ammonia-based fertilizers to the soil. Nitrification is usually of two types. First is the chemoautotrophic nitrification and second is chemoheterotrophic nitrification. Chemoautotrophic nitrification chiefly occurs in neutral to alkaline soils. This process is exclusively bacterial and is about 100 to 10,000 times faster than chemoheterotrophic nitrification. The later occurs in acidic soils and is carried out by diverse bacterial groups as well as fungi. An important distinction between chemoautotrophic and chemoheterotrophic nitrification is that acetylene inhibits chemoautotrophic nitrification, but it does not inhibit chemoheterotrophic nitrification. Nitrification occurs in two steps, and there are two groups of nitrifiers identified on the basis of reaction they catalyze. These are ammonium oxidizers, the names of which are prefixed with nitroso, like nitrosomonas, nitrosococcus, nitrosolobus, nitrospira, etc., and nitrite oxidizers, the names of which are prefixed with nitro, which include nitrobacter, nitrococcus, nitrospina, etc. The first step in nitrification is the oxidation of ammonium to nitrite, and second is the conversion of nitrite to nitrate. Ammonia monooxygenase first oxidizes NH4 to hydroxylamine, which is converted into nitrite by the hydroxylamine oxidoreductase. In the second step, nitrite is converted into nitrate by incorporation of oxygen from water. Nitrite is toxic even in small amounts. However, tight coupling of nitrification reactions prevents its build-up in the soil. Nitrification is a soil acidifying process, and acidification over a period of time inhibits nitrification. During nitrification, a small amount of the initial substrate, that is NH4, is oxidized to nitrous oxide and nitric oxide. Both these gases have global warming potential and also cause ozone layer destruction. The next process that we will be talking about is denitrification and dissimilatory nitrate reduction. Nitrate, after it enters the soil, can meet diverse fates, which include its incorporation into amino acids via assimilatory nitrate reduction, or reduction to nitrate, ammonia, or gaseous nitrogen via dissimilatory nitrate reduction. The reductive fates of nitrate are distinguished by their end products as well as by the ecological conditions in which they occur. Nitrate can be reduced to nitrate. The process is known as nitrate respiration. It can be reduced to ammonia. The process is known as dissimilatory nitrate reduction to ammonia, or DNRA, or it can be reduced to gaseous nitrogen. The process is known as denitrification. This depends on the species. All these are energy generating processes that occur in anaerobic environments. Respiratory nitrate reductases carrying out nitrate reduction are induced by nitrate in the absence of oxygen. Different types of nitrate reductases in microbial and plant world share one common feature, which is the presence of a molybdenum cofactor. Nitrate respiration is an important process in anaerobic metabolism. During respiratory nitrate reduction, nitrate is used as a terminal electron acceptor instead of oxygen. The energy generated in this process is utilized in the ATP synthesis and for maintaining cellular functions in bacteria. Denitrification is the process of removal of nitrogen from the soil in the form of a gas. Nitrogen is returned to the atmosphere via its conversion into molecular gaseous nitrogen, nitric oxide, and nitrous oxide through the process of denitrification. Denitrification thus causes the loss of nitrogen from the soil. The bacteria, Pseudomonas, and some fungi are chiefly responsible for denitrification in soil. They use nitrate as a source of oxygen in presence of glucose and phosphate. Denitrification of nitrate to nitrous oxide under anaerobic conditions produces 545 kilocalories per mole, whereas denitrification to molecular nitrogen yields 570 kilocalories per mole. Denitrification is favored in soils with ample quantity of organic matter to support the bacterial growth under anaerobic conditions. Loss of nitrogen can be considerable in warm soils that become saturated after 2 to 3 days. During DNRA, nitrate is reduced to NH4 and not to the nitrogen gas, as happens in case of denitrification. NH4 is effectively bound by soil clay particles, which prevent its loss from the soil subsystem. DNRA occurs in anaerobic environments like sediments and estuaries. DNRA to ammonia is an effective way to recirculate nitrogen in the system. Partitioning of nitrate to DNRA and denitrification depends upon the concentration of nitrate and carbon content in the ecosystem. In nitrogen limited and carbon rich systems, DNRA has been reported to remove more nitrate than denitrification, thus effectively circulating nitrate and preventing its loss from the system in the form of gaseous nitrogen. Now we will talk about the factors that affect the availability of nitrogen to plant roots in soil. The quantity of different forms of nitrogen in soil keeps on changing due to biological, physical and chemical processes. The availability of nitrogen at any given time depends upon the rate of processes adding and removing nitrogen from the soil. Factors that affect soil nitrogen content and its availability to plants include: first, soil nutrients. Nitrogen fixation holds key to entry of nitrogen into the living compartment of the environment. Nitrogen fixation is influenced by all those environmental factors which influence either the host or the symbiont. Apart from other factors, nitrogen fixation is strongly influenced by phosphorus content in the soils. It's usually accepted that nitrogen fixing systems require more phosphorus than non-fixing systems. Apart from general plant growth, phosphorus is required for the formation and development of root nodules. Next factor is soil moisture content. Soil moisture is an important factor affecting nitrogen fixation. Proper moisture content in soils is favorable for growth and development of plants and other soil inhabitants. Excessive soil moisture leads to waterlogging conditions, which in turn prevent formation of nodules and oxygen diffusion into the soil. Waterlogging also creates anaerobiosis, which favors denitrification. However, many nitrogen fixing species prefer aquatic conditions. Excessively dry conditions reduce the number of rhizobia in the soils and inhibit nodulation. Ammonification best occurs at 15 to 70% moisture content of soil pore space. Nitrification also occurs at soil field capacity. Next factor is soil organic matter. The availability of ammonium present in soil is significantly influenced by soil organic matter content. The amount of NH4 in turn affects the processes downstream. Organic matter serves as a source of carbon to denitrifying bacteria, which remove nitrogen from the soil. Next factor that affects the availability of nitrogen to plant roots in soil is soil temperature. Soil temperature influences the availability of nitrogen to plants in soil. Ammonification, nitrification and denitrification, all the processes involved in nitrogen cycling, are influenced by soil temperature. Now the soil oxygen content. The availability of oxygen in the soil profoundly influences different processes in nitrogen cycle in the soil. Some of these processes, like nitrogen fixation, are strictly anaerobic and must be highly protected from the inhibitory effects of oxygen on the enzyme systems involved in it. On the other hand, mineralization of organic nitrogen and conversion of ammonia into nitrate require oxygen availability. Soil pH is the next factor affecting soil nitrogen availability. Different processes in nitrogen cycling are especially sensitive to soil pH. However, nitrification occurs at a fairly wide range of soil pH, between 5.5 and 10. pH lower than 5.5 inhibits nitrifying bacteria, therefore adversely affect nitrification, which directly influences nitrogen availability to plants. Denitrification is reduced severely at pH 5, whereas pH 6 and 6.5 is optimum for denitrification. At neutral soil pH, nitrification is rapid. However, in acid soils, nitrification is slow. The equilibrium between ammonium in soil solution and ammonia gas in soil pores is significantly influenced by soil pH. Higher soil pH coupled with high soil temperature enhances ammonium volatilization from the soil. Now the volatilization. Volatilization is the loss of nitrogen from the soil in the form of ammonia gas. Ammonium is chiefly lost by this process, especially at higher soil pH. Loss of nitrogen from soil is considerable at soil pH higher than 7.5. Half of the ammonium in the soil is lost due to volatilization at pH 9.3. Volatilization losses are higher at higher soil temperatures. Soil drying after reaching field capacity increases volatilization loss as evaporation greatly favors volatilization. Now the precipitation. Nitrogen can be lost from the soil in the runoff, or it can leach down the root zone in solution form. Nitrate is highly water soluble and is lost with water that leaves the ecosystems as a runoff. Losses of urea and urea containing products as ammonia are greater in neutral to alkaline soils under conditions of a light rainfall. Now immobilization of nitrogen in the soil. Immobilization of nitrogen refers to its uptake by soil microbes for building up of their own cell mass. Thus, immobilization is locking up of nitrogen in microbial cells. Higher CN ratio favors nitrogen immobilization. Generally, organic remains with high CN ratio equivalent to 20:1 favor net mineralization, and those with CN ratio 30:1 favor net immobilization. Most of the well decomposed organic matter has a CN ratio of 10:1. Immobilization also includes a conversion of nitrogen into forms that would not be available for a plant uptake. For example, fixation of nitrogen into humus. Thus, immobilization refers to unavailability of a nutrient in the sense of a plant uptake. Fire is the next factor. Fire has been reported to affect soil nitrogen dynamics. However, effects of fire on nitrogen transformations in soil, including ammonification, nitrification, microbial immobilization, and denitrification, have not been adequately investigated. So, in this lecture, we first discussed the processes that affect the different forms of nitrogen in soil, and then afterwards we talked about the factors that influence the availability of nitrogen to plants in the soil. With this, I conclude this lecture and say thanks to you.